And the next question is, pesticide residues can be measured using which instrument? First of all, you should know what is pesticide residue. So pesticide residue is the leftover pesticides or its breakdown products which remains in the food, soil, water or plants after the application is done. So what exactly the pesticide residue means? When you spray a pesticide, it doesn't all vanish, right? So what remains is called residue. So the original pesticide molecule you sprayed, that is a parent compound. And metabolite is that the original parent molecule will undergo the breakdown. So that the metabolites will be formed from the parent compound. For an example, from DDT, DDE is formed. Then the degradation products also be occurring due to light, heat and microbes and all. Okay, and the pesticide chemically bound to plant or soil compounds. Those are not extractable easily. So that is called the pesticide residue. Okay, why do we need to check means that it should be below MRL, that is maximum residue limit. That is generally set by FSSAI and Codex and all. If it is above MRL means what happens? The food will be unsafe for the consumption. So for an export, the particular product will get rejected. Okay. So in order to measure this pesticide residue, which instrument is commonly used? They asked. The option given for this are GLC, that is gas liquid chromatography. Okay. Next, flame photometer, mass spectrometer, then calorimeter. And the correct answer is GLC, which is gas liquid chromatography. It is one of the commonly used analytical technique for detecting and quantifying pesticide residues in most of the matrices like food, soil, water, and even in the plant materials also we can detect the pesticide residues. What usually is being done by the instrument is that it separates the chemical compound based on their volatility, and also the interaction will happen with the liquid stationary phase. Okay, so this technique is commonly used for detecting most of the insecticides like organochlorine, organophosphate, and pyrethroids and all. Why it is having more sensitivity towards those insecticides, and also the result will be more accurate. So GLC is the standard answer for the pesticide residue estimation. And why not flame photometer? Because it is only used to estimate alkali like sodium, potassium and all, not for detecting the pesticide residues. And why not mass spectrometer? Means mass spectrometer generally identify the compound based on their molecular mass and fragmentation pattern. It is generally coupled with the GCLC and all. It is not used alone. So although it is having the advanced technique, the direct answer is GLC only, not mass spectrometer, because the detection of ions will happen by using this instrument, not able to identify or separate the compound. Okay, and why not calorimeter? Means that the calorimeter generally measures the concentration of substance based on the color intensity, which is used in biochemical estimation and nutrient analysis and all, not for pesticide residue analysis and all. Okay. And the next question is vector of Bhendi vine clearing disease. So the options given for this are aphid, leaf hopper, mealybug, and whitefly. And the correct answer is whitefly. So Bhendi vine clearing is caused by the vector, which is transmitted by the vector whitefly, because this will transmit the Bhendi yellow vine mosaic virus, that is BYVMV. Okay, so mainly it carries the virus belongs to Begomo virus. Okay, and the symptoms, how it will be looking like, the vines of the Bhendi leaf become clear or yellow in color, and the intervinal areas will remain green in color. and the whole plant look like stunted appearance, and there will be, fruiting loss occur. Reduced fruiting will be there. Okay, it's because this whiteflies are the primary vectors of most of the begomoviruses in Bhendi, tomato, and cotton and all. So they usually, transmit the virus in a persistent manner while feeding on the phloem sap. And aphid, this is a vector for most of the mosaic viruses, like, that is not for bendy vine clearing disease. This will be for Bungy top viruses, copy mosaic viruses and all, not for bendy vine clearing. And leaf hopper, this is the vector for phytoplasma diseases, and rice tungro, and not rice tungro is mainly transmitted by green leaf hopper. Okay, and this will be the vector for the most of the phytoplasma diseases, not for the bendy vine clearing. And mealybug, this again is the vector of wilt of pineapple and cocoa swell and shoot viruses and all, not for okra. So bendy yellow vine mosaic virus means that is mainly transmitted by whitefly. And the next question is, flat fan nozzles are exclusively used for spraying. Which one? Insecticides, fungicides, herbicides, or all of the above? What is nozzle? So nozzle is a very critical component which automates the liquid into droplet. Okay, and that helps to direct spray and controls the flow rate. It is like a small equipment which will be fitted in the sprayer. Okay, so the nozzles types are different. For each nozzles, there will be the specification given. Okay, some of the nozzles are useful only, they are meant to spray the insecticides, and some of the nozzles are for fungicides, like that. There will be the different types. So here the question is asked, flat fan nozzle is exclusively used for which spraying? And the correct answer is herbicides, and also known as fungicides. So remember one thing, flat fan nozzles, which is otherwise known as fishtail nozzle. Okay, so this nozzle is used for spraying weedicides or herbicides. Okay, if the question is asked like hollow cone or hydraulic nozzle means, the answer would be the insecticides or fungicides. Okay, so remember one thing, for insecticides and fungicide spraying, the nozzle type would be different. So for this spraying, mainly hollow cone or hydraulic nozzle would be used. Okay, and another nozzle is there, that is solid cone nozzle. Solid cone nozzle is mainly, used for spot spraying. So if there is a particular spraying like spot is required means, in that area we should use solid cone spot spraying. Okay, so that's it for this. Flat fan means, here the answer is herbicides. And the next question is, a biocontrol agent used to control Salvinia molesta. So the some insect would be used to control the weed. Salvinia molesta is the water fern weed. Okay, so the common name of Salvinia molesta is water fern. That is the aquatic weed. So in order to control this aquatic weed, there will be the specialized biocontrol agent used. What is that, they asked? So the options given for this are hyacinth moth, flea beetle, or else we can tell flea beetle, curculonid weevil, scale insects. And the correct answer is curculonid weevil. So see, weevils, beetles and all generally belonging to the family Curculonidae. Okay. So Salvinia molesta, actually this weed is most notorious weed. Why means that it is a fast growing free floating aquatic fern. Means it is an invasive weeds. Generally it can be seen in lakes, ponds and irrigation channels and all. and it will be forming the thick mats in the water. And since it is a forming thick mats, it will be, completely blocking the sunlight, oxygen, and water flow and all. So thereby, many organisms, aquatic organisms, will get affected because of this fast-growing aquatic fauna. So in order to control this, the most successful biocontrol agent is Cryptobagus salvinia, or we can tell Cryptobagus singularis, which is the curculionid weevil. It is generally used to control the water fund because this particular insect is native to Australia. So from there it was imported just to control the weed water fund. How by using this insect the weed will be controlled? You may be asking question. See, both larva and adult of this particular insect will feed on the Salvinia molesta, buds, stems as well, and roots also. Most of the parts of the Salvinia molesta will get completely devastated by this larva and adult of pest. It is not a pest, it is a biocontrol agent, it is a beneficial insect. Okay, since it feeds the plant, it will cause the plant to sink and die. So see, this particular thing is that host specific. Host specific means this particular insect will feed the Salvinia molesta only. It is a host specific insect. So it doesn't attack other plants. Okay, and it is used as a biocontrol agent successful in India and Australia and Africa and all, just to control Salvinia molesta, that is a water fan. And why other options are wrong means hyacinth moth, which is the Nymphograpta albicutalis, generally used for control the water hyacinth. That is Icornia crassipus. Icornia crassipus and all you may be come across, right? And those weed control this moth will be used. Flea beetle is used for controlling the alligator weeds. And scale insects means mainly that is used for controlling prickly pear. Okay, cactus opensia control, prickly pear, not for aquatic weeds. Okay, so prickly pear there is one weed that is opensia delinea. So in order to control prickly pear, this scale insect would be used. That is Dactylopius opensia. Dactylopius opensia, which is nothing but a scale insect. This insect looks like a scale. This is, imported from US. Main origin is US. Okay. And also remember one thing, to control Parthenium histophorus, that is Parthenium weed, you may be knowing about the weed. That is Congress grass and, carrot weed and all, we can call it the different names for that. So in order to control Parthenium, we can import the like Psychochroma bicolorata. Psychochroma bicolorata is the insect belonging to the beetle family. Okay, the main origin of the insect is Mexico. So Psychogramma is used to control the weed Parthenium, and Dactylopus opensia is used to control the weed prickly pear. And this Cuculonid weevil, that is Cryptobagus salvinia, is used to control Salvinia molesta. That's it. And the next question is actions to preserve the natural enemies. Natural enemies means it could be the predators or parasitoids. Okay, by environmental manipulation is known as. So by doing some environmental manipulation, we are going to save the natural enemies. So which action is preferable, they asked. So option A is augmentation, option B is importation, option C is conservation, and option D, which is a multiplication. And the correct answer is conservation. What is meant by conservation? So conservation means that the modification of environment or the existing practices to protect or enhance the specific natural organisms. to reduce the effect of pest. So that is called conservation. We are just going to create the different environment just by modifying something. For an example, conservation practices which include we can reduce the pesticide use. That is also one of the important conservation usage we can tell. And we can also, provide the flowering plants as a nectar source for getting the good survival of natural enemies like predators and parasitoids and all, because they are mostly relying on to get the nectar sources as their feed. And we can protect the natural enemies habitat. Okay, it is already the existing habitat will be there. We should protect them just from the pesticide spray and all. Okay, so that is the thing. So the correct answer is conservation. What about augmentation? So see, augmentation is different. Augmentation means where we are going to increase the population of natural enemies just by propagation or release. Either we will be multiplying it, or we can release the natural enemies. How it can be done? It can be done usually by two means. One is inoculative, another one is intentative. Okay, so that is having the deep subject. So as of now, you should know what is augmentation, just to increase the population of biocontrol agent, or else we can tell the release of biocontrol agent in the field. Okay, and what about importation? Importation is nothing but comes under the classical biocontrol agent. Classical means the old biological control approach, which means the introduction. So see, if something is not able to, having the control means in our area, like if something is causing the devastating effect means, in order to control that, we are going to introduce or import something from the foreign, right? So that is called importation. Okay, so it is like, generally the permanent establishment. For the permanent establishment of natural enemies, we are going to introduce something from the foreign countries. Okay, for an example, some pests are there in our country, like some pest, cotton cushion scale. You just take the example, cotton cushion scale. It is causing a devastating effect. In order to control the particular pest, the Vedalia beetle from US and all, California and all, can be imported. So this will be feeding the cotton cushion scale. So that is called introduction or the importation, and also we can tell classical biological control. Multiplication is just about the multiplying the biocontrol agents. That for an example, trichogramma and all can be usually multiplied in the laboratory conditions in order to, control the sugarcane internal border and all. Okay, so that is totally relevant to this question. So the correct answer is conservation. The next question is, which of the following organisms are not storage pest? Generally, what is a storage pest? Those organisms which damage the storage grains, seeds, pulses, as well as the processed products, whichever being kept in the warehouses and big sized godowns and all, right? So the options given for this are insects, rodents, nematodes, and fungi. And the correct option is nematodes. So nematodes are not the storage pest. See, insects are the main storage pest. Okay, not a nematodes. Nematodes primarily, can be seen in the soil and the plant, not in the storage godown. They usually attack root stems, leaves, whichever in the soil, right? And they don't even survive in the, dry stored grains and all. They need some moisture conditions. So they are not considered a storage pest. Okay, and insects, rodents, and fungi and all, we can tell they are the one which usually occurs in the storage godowns. Okay, and the next question is about Insecticide Act. So the Insecticide Act, enforced by the government of India during 1968, regulates import of pesticides, manufacture of pesticides, research on pesticides, and transport of pesticides. So above these four answers, which one is correct? They asked. So the different options are given, like 1, 2, 3 are correct, and 2, 3, 4, like that different options are given. So before solving this question, what you should know? You should know about the definition of the particular act, Insecticide Act, which is regulated in 1968. What this act tells about, that you should know. And the correct answer is import of pesticides, manufacture of pesticides, and transport of pesticides. Because this act is mainly enacted to regulate the import, manufacture, sales, and distribution, as well as transport, and also the usage of insecticides. So remember one thing, this tells about six main things. What are all they? Import, manufacture, sale, transport, distribution, and use of insecticide. Okay, so if these six are given as options, means you can opt it. But here, relevant to this, they have given research on pesticide. So this is totally irrelevant to the question, irrelevant to the Insecticide Act. Okay, and also this act main objective is to prevent the risk to whom? To human beings as well as to animals. Okay, in order to ensure the pesticides are handled safely. Okay, so the options are import, manufacture, transport. If they are given sales and distribution and usage of insecticides, means that also you can include it. Okay, and the next question is, airy silkworms are commercially reared on. You might be heard about this airy silkworm. There are different, silkworms are there. What they are? Mulberry silkworms are there. Tropical silkworms are there. Tassar silkworms are there. Mukha silkworms and all there. So different silkworms are there. Okay. In our condition, in the Indian condition, what mainly we are giving the preference to means mulberry silkworm, right? So here the question is asked, airy silkworm. Airy silkworm is usually reared on which leaves, they asked. Option A, mulberry leaves. Option B, castor leaves. Option C, tobacco leaves. Option D, none of the above. And the correct answer is castor leaves. Because, airy silkworm, which is Samia zinthia resini, is commercially reared on castor leaves, that is Ricinus communis. Okay, and also there will be the sync between the scientific name of these two. Airy silkworm scientific name is Samia cynthia resini. There also resini is there. What is the scientific name of castor leaf? That is Ricinus communis. Here also resin is there. So in that way we can correlate this. Okay, so this airy silkworm is the polyphagous insect, but castor leaf is the primary host for commercial rearing in India. Okay, and also other secondary host reported, those are kesaru, tapioca, and all. But the primary host for the airy silkworm commercialization is castor leaf. And mulberry leaves is mainly for rearing the mulberry silkworm, that is Bombys mori. Okay, and tobacco leaf is not used for any silkworm, like any commercial silkworm rearing. Okay, and none of the above, like you can choose. If nothing is related to the question, means you can choose none of the above. Okay, so castor leaf, that is mainly preferred by airy silkworms, and also you should know the airy silk. So the commercialization on the airy silkworm is airy culture, right? The particular study is airy culture. Airy culture means that mainly the airy silk, which is reared from this, are called airy silk. Airy silk is also known as Endi or Erandi. Okay, the other names of airy silk, Endi or Erandi, which is usually white colored one. Okay, and mainly this airy silkworms, reared and producing the airy silk is majorly prevalent in northeastern states. northeastern and Bihar, West Bengal, Orissa, where you could see this commercialization is quite popular over there. Okay, and mulberry leaf, that is mulberry silk. So the product from this rearing is called mulberry silk. So finally, we are going to get the silk right after the weaving step is done. They will, extract the silk from the cocoon of the particular insect, right? So the egg will be there, larva, then cocoon, then adult. So from the cocoon, they will, try to extract the thread that is called silk. So from the mulberry silkworms, they will get the mulberry silk. Okay, so that is quite popular in Karnataka and Andhra Pradesh and all. And one more thing is that is Muha silk, which is very important. Using the Muha silkworms, they will get the Muha silk. Muha silk, mainly commercialized by rearing the Muha silkworm Antheria assama. So that is Satinide butterfly, the big sized, that is a moth-like insect, big size moth-like insect. Usually that will be reared on Som and Solu, the host plants. And mainly that is prevalent in Assam. That is particular to monopoly. Monopoly means that because that silkworm is particular to the Assam area. Okay. And one more silk is there, that is Tassar silk using Antheria milita. Okay, that is, commonly prevalent in Jharkhand, Maharashtra, West Bengal and all. So there are 5 different silks are there. Mulberry silk, then Eri silk, then Muha silk, then, tropical Tazar silk and oak Tazar silk. Okay, oak Tazar silk is another type that is commonly prevalent in, sub-Himalayan belt. That is mainly, reared on Kurkus species. And the next question is the chemical responsible for the production of silvershoot in the rice due to the attack by rice gall midge. So they said about the particular chemical is produced by the rice gall midge. But which chemical they asked? So due to the particular chemical in the rice, there will be the formation of silvershoot. Okay, so option are given for this: cumanin, cecidogen, orazinone, phenol. So the correct answer is cecidogen. Okay, this is the chemical produced by this rice gall midge, Orciola oryzae. So because of this attack, the growing point of the rice will get killed. Okay, and infested tiller will become, a tubular in structure. They will be having tube-like structure, the infested tiller. And silvery white and onion, it will be like onion leaf. So silvershoot is also known as onion leaf. Silvershoot symptom or onion leaf symptom is mainly produced by rice gall midge. So which causes the major, yield loss in rice. Cecidogen is the chemical, or we can tell growth regulating substance, mainly secreted by rice gall midge larvae while it is feeding. So this particular chemical alter the normal growth of the rice plant tissue, especially at the growing point, so that it will be causing the tubular like structure in the infested tiller. And also we can tell that is onion leaf like structure. And this will lead to, abnormal cell elongation and formation of characteristic silver shoot. It is essentially the gall inducing chemical mainly produced by rice gall midge. Okay, and cumanin is basically a phytoalexin which is produced by carrot in response to fungal infection. So this has nothing, to relate with this question. And oryzonenone is like it's not a standard chemical associated with the rice gall midge. Oryzanol is a natural, antioxidant mainly found in the rice bran and bran oil. It has nothing to do with the silvershoot production in rice. Okay, And phenol is again, it's a general, it's very general phenolic compound, not a specific gall producing chemical. Okay, so the correct answer is acidogen.